Great. So yeah, as Rich said, I've been set up great for my talk. A lot of what's been said already is I'm going to build off of. So yeah, these have been really interesting so far, and it's great to be a part of this. So if you just go to slide one, advance one slide down, I'm going to talk today about how management affects stormwater biofilter function. And really, I'm just going to focus on harvesting the biomass. So you could see here a picture of one of the biofilters in a suburb of Melbourne, actually in the Little Stringy Bark Creek project. This one is at Morrison Reserve. It's a pretty large biofilter with a ponding zone that's been mowed. And so this biofilter's biomass, the above ground biomass, has been cut and removed. And so there's a few things to this problem, or to this, I wouldn't call it a problem, but to this management technique that we really don't know much about. Partially, it's that we're removing these nutrients that are bound in the plant biomass, so that might have an effect if the plants were to die back on overall nutrient removal. But then you have plants that don't have as much leaf mass, and so they're not photosynthesizing as much, and perhaps not transpiring and affecting the subsurface biogeochemistry, or taking up as much nutrients. And so I'm going to go over a couple of the problems and the pros and cons of harvesting the above ground biomass. So if you go down to slide 2, start in first with, I'll give you the bad news before I give you the good news. One potential problem with harvesting is that when you take the top off of the plants, you're potentially reducing the gas flux between the subsurface and the atmosphere. And so that has pretty big implications for removing nitrogen gas that's just been denitrified in the subsoil in that rhizosphere. Because the plant isn't transpiring as much, it might not be passing as much, excuse the phrase, but passing as much gas to the atmosphere. And so there was a study by Jay Martin at Ohio State that looked at different harvesting rates, and they found that the more a plant is harvested, this is in a constructed wetland, the less gas flux occurred from the subsoils, particularly in the rhizosphere, to the atmosphere. So it actually had decreased, in the constructed wetland, decreased the nitrogen removal, because this reduced evapotranspiration. So that could be a possible problem with harvesting the biomass. If you go down to the next slide, slide 3, another one is that this problem that could occur if the timing is not right. So we've seen, I'm going to talk a little bit about some of the maintenance schedules we've seen for these systems, but one interesting thing out of a study out of, I believe it was in the somewhere in the Midwest, looking at TIFA for biofuel production, and they also wanted to see if they were able to optimize nutrient removal by the timing of their harvest. And what they found was that in the winter months, so this is in the Midwest and in temperate climates, late to midsummer is when most of the nutrients in the plant were concentrated in the above ground biomass. And following that period, in the fall, the plant started to translocate the nutrients down to the rhizomes. And so if they had cut the above ground biomass following that translocation, they would have missed their opportunity to collect a lot of the nutrients that were incorporated in the biomass during the summer. And so timing is really important. On the flip side of that, if you look at Mediterranean climates, our dieback is typically in the summer. So there needs to be a little bit more thought to as the optimal timing of harvesting in different climates. Right now, there's not a lot of research on that. So another issue, if you go down to slide 4, is that phosphorus really isn't all that affected by plant uptake in biofilters. If you look at this diagram from a study done in Queensland, these are two different plant types. The B and the S stand for the plant types, and then the H and the N stand for harvested or non-harvested. I use a similar naming convention in the research I'm going to present later. And what you could see is a partitioning of phosphorus, and the up arrow is the above ground biomass, the down arrow is the below ground biomass. You see when with the harvested plants, there's a lower nutrient content, a lower phosphorus partitioning in the plant tissue in the plants when they were harvested than when they are not harvested. But the real important thing here is that number in the lower left corner of each of those boxes, the 98.1, the 97.7, those are the percent of total phosphorus in the soil part, the soil pool. So really, like a big problem with a big issue with constructed wetlands is this phosphorus removal really isn't driven as much by the plants as nitrogen removal is. And so removing the above ground biomass of plants isn't as advantageous for something like phosphorus. You go down to the fifth slide, and I talk about species-specific responses to harvesting. So different species react differently to being harvested. And not all species are really adapted to being grazed or harvested very well. So basal monocots, basal meristem monocots like grasses and sedges and rushes and things like that are typical in grasslands were evolved with grazers, and so they have their meristem at the near the base of the plant, and so when a lot of times those regrow a lot faster than something like a woody shrub or a tree, and so cutting off the tops of those usually produces more regrowth faster. But even similar types of monocots like the sedge and the carex here in the Phacenia that I show, the carex actually regrew quite a bit faster and replenished its biomass faster than the Phacenia in our study. And I'll show those data in just a little bit. One of another issue with harvesting, you can go down to slide 6, is just the logistics and costs. So we've heard from almost every talk now about the aesthetics, and so that's really important. A really important part of maintaining the aesthetics is harvesting and taking away the dead leaf matter. But here we see on the left side, in August, at one of my favorite biofilters in the world, Edinburgh Gardens, has a big problem with sediment at the inflow, and then the plants aren't really growing around it, and the plants that are there, even though it's towards the end of a wetter period, they're not really doing all that well. There's a lot of dead material. So they went in early October, the photo on the right side, and they harvested all the plants and then put in mulch. So that's a pretty big project. This is a big biofilter. And so just hiring a company to do that knows how to work on these biofilters, and then the actual cost of the work involved is a pretty big undertaking. If you can go down to the next slide 7, you can see the progress of this, though. So even though it is a potential problem with harvesting, the logistics and cost, it is paying off. So by July of 2015, a lot of those plants have reestablished after being harvested and even in some cases replanted. I do have a maintenance schedule here and some email correspondence saying that this biofilter, one of the entire cells, was replanted, which is interesting. If you get on to the next slide, this is the actual maintenance schedule, and I'm on slide 8 now, of Edinburgh Gardens. And so these two circled in red highlighted line items are just for maintaining the vegetation. So you can see the harvesting occurs every 3 months in this case. And so that was really interesting to see that they actually do this a lot more frequently than I expected. And they're going out monthly for other things, so they actually may harvest more than that, and they definitely remove debris and litter more often than that. So if you go down to the next slide, slide 9, we'll get into the sunny side of harvesting. So one good thing about harvesting is that the plant uptake could potentially increase whenever you harvest, because that new shoot growth a lot of times and a lot of species of plants actually has greater nutrient content, and that growth rate is also usually a lot faster than the growth rate prior to harvesting. Now if that deficit is is taken up is actually met by the decrease in photosynthetic activity is something that you have to look at over a longer time period. But that's potentially one of the benefits of harvesting biomass. If you go down to slide 10, in some climates there's pretty significant die-off, and so those decomposing and the mineralizing nitrogen and phosphorus in the plant matter would then be reintroduced to the system if it wasn't harvested before the die-off occurs. If you go down to slide 11, you can maintain a ponding zone by mowing. So this is one of the first biofilter pictures I showed, Morrison Reserve, where the ponding zone was previously clogged or blocked, not clogged, but blocked by the a lot of vegetation in the ponding zone. And so keeping that ponding zone clear of debris can be important for the function of the biofilter. But then the next slide, slide 12, I want to get really to the most important part of this, which Rich touched on, is the aesthetics. And I gave the example earlier of Edinburgh Gardens. They harvested and replanted, but a big part of this was harvesting to maintain the plant health, and that really maintains the look of these things. And this biofilter is in Edinburgh Gardens is a really popular one. I mean, I don't know that a lot of people know what it does. But there are always, on nice days in this park, there's always a ton of people around it. So they're looking at it all the time, and so it's a nice project to keep looking nice. Another aspect of harvesting is to maintain sight lines. So for curb cutouts, if you're driving in your car, you don't want to be looking over your shoulder. You can't see anything past this big nuisance of a biofilter if the plants are growing too high. So a lot of times they have to cut down plants if they're not selected correctly to be low-lying plants. They actually had to harvest those on curb cutouts on the sides of the roads. So we were kind of thinking about these aspects of management, and so we wanted to see how this harvesting actually affected the function. So if you go down to slide 13, we asked the question how harvesting affects stormwater runoff treatment, and we were looking at mainly nitrogen and phosphorus, and I'm going to talk about nitrogen today. So we did a column study at Monash University with two of the most common biofilter plants we encountered, and that's Carex suppressa and Phocinia nodosa, and we harvested half of them and didn't harvest another half. Excuse me. If you go down to slide 14, you can see a diagram of the columns that we built. There is a saturated zone that's supported by the raised outflow on the left side. That is to ensure that the water level is maintained at a higher point than the bottom, so these roots are able to access a saturated zone, and that can help with denitrification through the through an aerobic and anaerobic oxidized zone in the rhizosphere in the submerged zone. And then a typical filter media, which is a loamy sand, is on top of that saturated zone, and that's the top 300 millimeters of this system. And then they were planted with either Carex or Phocinia and allowed to mature for 20 weeks. They were dosed for based on the mean annual rainfall divided up to two times a week doses. And the synthetic stormwater recipe we used was based on typical Australian stormwater runoff. So it had nitrogen and phosphorus and metals and some stormwater sediment too. So the amount that we dosed was equal to a drainage area that's about 50 times the size of the area of this biofilter column. So it ended up being something like 8 liters a week for each biofilter that we added. We measured water quality 10 weeks and 2 weeks prior to harvesting, and then 3 times following the harvest. And so this is just a picture of the planting that was done. So we washed the roots free of the potting soil mix and then planted it in the columns. And so if you go down to the next slide, 15, this is just to show you that all of the biofilter treatments that were planted, we also had an unplanted control, which is the UN treatment. All of them were performing very well for nitrates, so the more than 90% removal for nitrate, total nitrogen and phosphorus, which becomes an issue when you're trying to detect the significant differences between treatments, because something, if it's already removing things as good as it can, something has to get worse for you to detect the difference in the treatment. And so we were hopeful that the biofilters actually didn't improve in treatment efficiency. But if you continue down to slide 16, just so you can go past that, this is a picture of the harvested biomass, and then a picture of the two columns that are in the same block that were harvested and not harvested on the right. So slide 17 shows growth rate, and so this is based on the difference in the average height of the tallest 5 stems over between sampling periods. And so you can see in the dotted lines following the pruning, the date of pruning, that we had a huge increase in growth rate in the carex, not so much in the Phacenia. If you go to slide 18, you can see that in the light green portion, the medium green portion, so that's right above the dark green on slide 18. In FH treatment, that kind of thin section of medium green is a lot smaller than the kind of thick section of the carex that was harvested. If you add back the harvested above ground biomass to the final biomass of these after the column takedown after our experiment was over, that's after 10 weeks of regrowth, that there was no significant difference in the final above ground biomass between the harvested and non-harvested treatments. If you go down to slide 20, I wanted to show a few of the major nitrogen pathways, and you can just kind of think of it. I'm not talking about, sorry, I'm on slide 19 now. I don't know why I said 20. So if you look at the major nitrogen removal pathways slide, most of the nitrogen that's leaving is through denitrification or plant root uptake or the effluent. So the nitrogen that's coming in the stormwater dose is really either being taken up by plants or denitrified and either passing through the plant roots and then into the plant stem and then in the atmosphere, or through the soil pores, and then some of it is coming out of the effluent as well. But if you take away the plant in slide 20, then you could possibly not just decrease the plant root uptake, which it makes sense because there's less photosynthesis happening, less uptake of water into the roots, and so you're probably taking up less nitrogen, but also likely less denitrification because that coupled oxic-anoxic zone around the rhizosphere of the roots is not as strong as it was when the plant was transpiring more. And so if you go down to slide 21, I did a little mass balance of the nitrogen over that 10-week regrowth period, and so you could see that total amount of nitrogen that removed from the biomass in the carex was about 100 milligrams of nitrogen more than in the Phacenia. And then the carex continued to improve nitrogen removal, or continued to remove nitrogen more than Phacenia following harvesting. So that's shown as the red section above the blue section. And so you can see there was 125 milligrams of nitrogen removed from the stormwater following harvesting in that 10-week period. And then there was only 113 milligrams of nitrogen removed in that 10-week period in the Phacenia that was harvested. And then the box with no fill in it, the 124 milligrams of nitrogen was the carex and Phacenia that were not harvested. Both over that 10-week period removed about 124 milligrams of nitrogen. And There is a significant difference if you go down to slide 22, and the Phacenia that was harvested actually had less removal of nitrate than its non-harvested counterpart, whereas Carex had no effect. So there's a species effect that's happening for Phacenia for harvested treatments. But it's important to note here that this reduction is still really high. So it's still 80 to 90% reduction in nitrate. If you go down to the next slide, total nitrogen is removed. So this on slide 23. You have a similar pattern, and so it's likely that the reduction that happened occurred because of a reduction in denitrification. That's the removal of nitrate. So this total nitrogen reduction in treatment from the harvested Phacenia was likely because the Phacenia was not removing nitrate as efficiently as its non-harvested counterpart. And this is still to be verified by some of the data I haven't analyzed yet, and that's the label nitrate data. So we're still looking to verify some of these things. So I just want to conclude on slide 24 that harvesting biomass is a really common management practice. We don't really know what it does to biofilters and their function, particularly with nutrient removal, and we didn't even talk about metals yet. In our study, harvesting the biomass did not decrease the total nitrogen removal drastically in the first 10 weeks of regrowth, but there was a species effect, and Carex was unaffected by harvesting in its removal efficiency, but Phacenia was, which is likely due to decreased denitrification. And multiple seasons and more species should be investigated because this practice is really common, and we're really not sure what kind of species, how different species react to it. So thanks for your attention. Slide 25 just shows our setup, where Andrew and I spent a lot of time collecting data from these columns. And thanks for your attention. All right, I'll take questions now.
All right, yeah, thanks very much, Brandon. So anybody have questions for Brandon?
Yeah, I do, Brandon. Great talk. Thank you. It was nice to see that data summarized so cleanly. I'm wondering, did you have a chance to see? I think you did. Perrin Cook's presentation yesterday.
Yeah.
Yeah, and so I was just kind of wondering if you could reflect a little bit on mechanisms. So one of the things I think he focuses a lot on is the fate of ammonium in these systems, and so basically looking at coupled nitrification, denitrification, and you know, like for example, when I was looking at your nitrogen sort of cycling within the biofilter, I noticed that there was not production of ammonium by ammonification, respiration of the organic material within the biofilter. And so that would be, you would essentially have an in situ source of ammonium being generated within the biofilter by ammonification. And then you have the ammonium coming into the biofilter from the synthetic stormwater. And I'm wondering, you know, really fundamentally, what is the relative concentrations of where's the nitrogen in the stormwater? Is it in ammonium? You know, is it in nitrate? And what are the relative importance of the various pathways that you know, that Perrin talked about: coupled nitrification, denitrification, direct denitrification, and ammonification?
Yeah. Okay. Good question. I didn't present ammonia concentrations because there wasn't a big difference in the treatments. The only difference was that Phacenia outperformed Carex, and that was harvested. And other than that, there were no really significant differences, except everything did better than unplanted. What I can tell you about the relative concentrations is that ammonia is about 0.3 milligrams nitrogen per liter. Nitrate, and this is in the inflow. Nitrate is about 1 milligram per liter, and then total nitrogen is like 2. So you have about a point, I guess it's 0.7 or 0.8 organic nitrogen as well. And so I didn't put the, I only wanted to talk about the removal pathways for this this talk because I was looking more at doing a mass balance of the column, more black box. So I didn't include transformations within because the main removal pathways are really through this denitrification, the nitrogen gas leaving through the plant stems, and then the plant uptake that is then either over a period of time captured in the plant biomass or removed when it was removed during harvesting. So the ammonia that's in the incoming stormwater runoff being transformed has an end product of either, in a lot of cases from what we saw was either in the end product being nitrogen gas or in the effluent. There's some absorption of some of it staying in the soil, but yeah, so I was looking at it more from a kind of a black box scenario.
So just to follow up then, so it sounds like the runoff is very similar to what Perrin described for streams, basically that they're nitrate-dominated systems. Mm. And so direct denitrification, or in the case of plants, plant uptake, are really the two competing pathways that you would have to focus on. And your results then, do you think they're consistent with Emily Payne's conclusion that when the plants are there, it's really just about, it's mostly about uptake and assimilation, it's not about denitrification? Or sorry, that was supposed to be one of the major conclusions, I just missed it, but.
Oh, no, yeah, no, the two main ones, I'm not sure that I can look at the data right now and tell you that which one is the main one, but the two main ones are denitrification and plant uptake that I expect to be the major nitrogen removal pathways. Like I said, I haven't looked at those labeled nitrogen data, so I'm not sure in what part of the biofilter the nitrogen is ending up, and I'm not sure if it's in the plant biomass that was removed. So those graphs that I showed was based on average sedge concentrations of nitrogen, not on the actual data. So there's still some work to do for telling how much, determining how much nitrogen is removed in the biomass.
I have a question about the harvesting.
Yeah.
Since it's clear that some managers are doing harvesting, do you know what criteria they use to determine when, where, and how much?
I think it's mostly based on landscape architects or gardeners evaluating the health of the plants.
So it's not like based on an effort to maximize the function of the biofilter in removing nitrogen or contaminants or whatever.
From what I have seen, nobody's talked about that from a maintenance standpoint for the biofilters. And if anything, the only thing I could look at that I could tell from the reason why they would remove biomass for function is for maintaining that ponding zone, the volume of that ponding zone, keeping it free of debris.
But in theory, your work would allow them to be a little more motivation-driven and.
Yeah, I mean, I think it's really useful to show that harvesting the biomass, if it shows that it doesn't really decrease nitrogen removal efficiency very much. I think that's very useful to people. So, but if it did reduce in some plant species, then that could be important, and they could that could be used from a management perspective to not plant certain species in areas that will be mowed, that need to be mowed to maintain the aesthetics of the biofilter.
So just, sorry, go ahead.
Oh, just a quick question. You know, I thought that was really great. And a lot of times, you know, when we present these results, sorry, we talk about how you know there was a significant difference in the outflow, but the treatment was all greater than 80% removal. So, and it's like, okay, so maybe it doesn't really matter. But I was thinking, I mean, these are small columns, and I mean, what if you had a biofilter with a really large surface area? Percent reduction may have been a small change, but could the load that's coming out be really large?
Yeah, that's a good point, because sometimes these things are completely mowed, and like the one I showed was just the ponding zone, and it's really hard to get a sense of how these things are managed, because it's so different with every different municipality and even system, that really how they're managed and how they're harvested or mowed is kind of really difficult to quantify, and so making recommendations, you could, I guess you could make more recommendations on the plant selection instead, to get at what you're saying, that mowing all of it may actually have a big impact in that reduction, as opposed to like a small area. And so I mean, I think this goes to making recommendations for really plant selection, maybe, rather than the management, if they're going to mow them anyway. But, yeah.
And sort of just a follow-up question, and maybe this isn't a knowable thing, but so you're taking the nitrogen out of the biofilter, and you know, sort of, I guess my first question is, where are you putting it? I mean, where does this material go, and what is its ultimate fate? And then the other question is kind of more, I guess, fundamental. It's sort of, if you were to think about the relative magnitude of the pools of nitrogen in your system, and you could sort of identify what those were, you know, the organic nitrogen maybe bound up in dead material within the biofilter, which accumulates over time, and then the roots in the plant above ground, and then nitrate and ammonium, inorganic forms, sort of like, what would be the relative, so where is most of the nitrogen, and how unknown do you think that picture is? So two-part question.
Yeah, well, first, with the harvesting, I imagine that it's in a lot of cases, most kind of bigger cities have composting services, so a lot of any kind of like lawn debris is probably going to go to the same place, which is the compost facility. I guess in some areas they might have, I don't know about for yard waste, but so compost facilities that then get the organic might get concentrated and then redistributed to yards and gardens and that kind of thing. So in a way, it's some sort of bit of a closed loop in that sense, that the yard waste that's occurring in a city is staying in that city through the redistribution of that compost. As far as where most of the nitrogen is in the biofilter, I think that probably a really large part of it is in that plant biomass, and it just kind of speaks to the complexity of these things, of how timing that harvest is really important to prevent the leaf litter from reintroducing that nitrogen back into the system. A lot of that, that leaf litter is probably removed through maintenance, though. So not just harvesting the plants so that they regrow and take up nitrogen again and go back to the same function they were, the same level of function they were before they were harvested, but also just removing that biomass that was there that contains what was previously taken up in that plant. It probably happens more than once a year in a lot of cases. I can think of most of the sites that we visited more than once, so that almost, not exclusively in Melbourne. I want to say well over half of them were managed by harvesting. So I can only think of a couple out of the dozen to 20 that we visited that we saw over the 2.5 year period that we were looking at these things were not ever harvested. And so I think it's a really pretty common practice. And so that's anecdotal evidence, but that's really, I'm thinking this is a pretty common practice.
Thank you, Brandon.
Can I ask one question for
Yeah.
Sweeton? All of the systems that you were looking at had submerged zones in them, yes?
Well, so just the column study?
Yeah.
Yeah, the column study had submerged.
So I was wondering if you could speculate on whether or not the story would be different if those systems hadn't had submerged zones. Would you expect a different effect of harvest?
Yeah. Yeah, that's interesting because I don't know if the health of the plant would have been as good if there was no submerged zone. And then that difference that I expect is happening because of perhaps decreased denitrification after harvesting, that difference might not have been there, so harvesting might have actually improved nitrogen removal efficiency even more because the denitrification wasn't affected because it wasn't there in the first place. But really, it just it needs to be done with a system like that instead. So some of that hopefully will be answered by looking at the labeled nitrogen data.
Okay, yeah, I was just interested, given that the systems we have here tend to currently not have submerged zones.
Right. Yeah, and if it is, if that difference is so minuscule that we're not really worried about how harvesting affects nutrient removal, then that's fine. Then we can suggest that people still go about mowing their biofilters or removing the biomass. Yeah.